All right, next up we have my colleague Joe Duris from the USGS, and he will be presenting on some of the CyanOHAB studies that we have going on within the USGS and the Great Lakes.
Thanks, Sam. So, Dr. Diaz, I'm gonna follow you. I can't stand behind the lectern. I can't do that. Thanks, Tammy, for the introduction. Thanks for having me here to talk today. So I'm gonna kind of take this because Amber just had such a great talk, and then we all asked her questions about, well, what about this piece of data and what about that piece of data? I'm going to kind of give an overview of the activities of the Great Lakes Restoration Initiative, the USGS Harmful Algal Bloom team. And we're usually pretty particular about this. We usually call them cyanoHABs when we talk about them. So they're cyanobacterial harmful algal blooms. That's specifically what we're interested in. The way that I'm going to go through the talk today, I'll kind of give a little bit of an outline, and then I'll talk a little bit about the processes and kind of how we think about things conceptually. And then I'm going to talk about the data that we collect, and I'll end with a few examples of some of the data analysis that we've done. So to move in first, I would be remiss if I didn't mention our team. This is not just me by a long shot. In fact, I'm kind of on the lower side of this. I manage a lot of the data, and I help to make sure that people have what they need. But these are the group of folks that really make this go. Marianne Evans is in our Great Lakes Science Center, which is in Ann Arbor, Michigan. Carrie Givens, Lisa Fogarty are in Michigan, and Pete Lineker is in Wisconsin, which is now part of our USGS Upper Midwest Water Science Center. So we're constantly getting shuffled and renamed. Erin Stelzer is in our Ohio, Indiana, Kentucky office. She sits in Columbus, Ohio at the Ohio Water Microbiology Lab. James Larson is an ecologist from USGS Upper Midwest Environmental Sciences Center. And Keith Lofton is he's an engineer by trade and a chemist by passion, and he works at our USGS Kansas Water Science Center at the Organic Geochemistry Research Lab. So as you can kind of tell as I went through, all those people have kind of very different views about these issues. Some people are really concerned with how the toxins are cycling and how they're forming. Other people are really concerned with bloom initiation, what's driving bloom formation, what's driving bloom senescence. It's one of the things that we don't think a lot about, but we're really interested in what makes it go, but we also want to know what makes it stop. And then some people, Carrie and Erin in particular, are very focused on the microbiology and the microbial interactions that are happening within these blooms. So this has been said, I think, in every talk, but I'll say the same stuff. We have these cyanobacterial harmful algal blooms. They've been occurring annually in Lake Erie, and they've been getting worse in the last 15 years or so. The severity of those fluctuates year to year. Severity can mean two things. It can mean the kind of extent, so how big the bloom is, but then also the density within that extent, so how dense the cells are there. USGS has been conducting research around the basin as part of the GLRI for a number of years, since about 2014. We're just in the process now. We feel like we've got enough of the variability hammered down that we're starting to evaluate data and look at it across multiple seasons. So where have we been looking? Well, primarily in the western basin of Lake Erie. We're there with about, I think, 5 other federal agencies and about 20 universities evaluating what's happening in the western basin of Lake Erie. But in addition to that, we felt it was important, because we were being funded by this Great Lakes restoration and not western Lake Erie restoration, to really make this kind of a broader Great Lakes study. The core of our team kind of sits in that central area in and around Michigan and Ohio. So we also evaluated the Saginaw Bay. That's not the right button. Laser, there we go. We also evaluated the Saginaw Bay. We evaluated the Grand Traverse Bay. And for a methods comparison, we evaluated Green Bay in Wisconsin. If you know anything about these bays that I just described, obviously we have a very eutrophic bay. We have a mesotrophic bay in Saginaw Bay, and an oligotrophic bay in Grand Traverse Bay. Green Bay is very seasonally eutrophic during the summer, much like the western basin of Lake Erie. It is thick with microcystis. What we did in these locations, we've been just doing like very routine monitoring down in the western basin. We did a trophic state comparison study in 2015 where we evaluated it and used the exact same methods on these 3 bays. And then again, as I said, this was a molecular method comparison that we did up in Grand Traverse Bay, or in Green Bay, because one of the things that we're very concerned with is you have a lot of people doing this research and everybody's using different methods, and we want to say what NOAA says is similar to what USGS says, is similar to what NASA says when we do all this information. So, a conceptual model. I like this model because it shows just how complicated it is, and it's in fact only really begins to scratch the surface. So it's a very simple cartoon. So what this cartoon indicates here is you can see you've got a succession of organisms in the water column, right? You've got algae, you might have some microcystis cells, you have some diatoms, and then with the addition and changes in temperature and carbon dioxide, in nutrients coming in and out of the system, and nutrients coming from different landscapes, you get this succession of bacteria and of algae and zooplankton and all of those things, and that comes into play. And you eventually get to a tipping point in the population where you get enough microcystis, that's your Cyanobacteria du jour, that's primarily what we're focused on, that you get a bloom forming. Okay, so once the bloom forms, that's not the beginning or the end of it. That's kind of the middle of the process in a lot of ways. Those of you who look at some of that buoy data, you might see that the pH changes on a daily basis. That's very driven by the photosynthetic processes that are happening within the bloom. As the bloom photosynthesizes, right, it's taking carbon dioxide out of solution. The pH then begins to go up in the lake. That has an effect on what metals are in solution, so it begins to affect solution chemistry. There's a lot of important things that are happening there. There's a kind of a fight for carbon dioxide as the bloom gets more and more dense. Dr. Diz and Dr. Campbell both mentioned temperature as being a very important driver of these things. You have to get to a certain threshold temperature before you're really going to see cyanobacteria take over. And then at some point, and for some reason, and I'll make that statement loudly, toxins are produced. One of my favorite ecological questions is, why do they make the toxin? They don't make it to poison dogs or to poison people. They certainly do it for some ecological reason. And there are a lot of hypotheses out there about why that is. One of those hypotheses is that this toxin actually might be a way that the cells are communicating with each other. Another hypothesis says that it potentially has something to do with metal sequestration, so that they can get rare trace elements that are hard to find and they need to scavenge in the bloom. Yet another says that it might protect them from over intense UV light, so it's some sort of large sunscreen molecule. All right, so there's a lot of ideas out there about what those molecules are. And then, of course, you have this bloom senescence, so maybe you get a viral infection, right? We don't think about that. The bloom gets sick. Maybe that's what causes the bloom to senesce. And then cells deposit. Some of those cells are alive, some of those are dead. And then you might get wave action that resuspends that back up, so you have this kind of ongoing cycling process. And then certainly there's other things that we need to think about. We need to think about nutrients coming out of the soil, right, or out of the sediment. When we get anaerobic conditions, we know that dissolved phosphate will oftentimes rise back into the system, and that can affect things. We also know that nitrogen can pulse out as ammonia from certain anaerobic sediments. We have the effect of secondary producers. So you guys might know this, there's 1 or 2 zebra mussels in the western basin of Lake Erie. We know that they filter the water, they clarify the water. That clarity leads to deeper photopenetration. It seems like, based on a few experiments that have been recently published, that zebra mussels and quagga mussels can preferentially feed on green algae, meaning they're choosing to eat green algae and spitting out cyanobacteria, enriching the cyanobacteria in the water column. And then external to that, we have these weather inputs, right? We've got days where it's sunny, we've got days where it's cloudy, we have days where there's wind, where there's stream inputs that are really high. So all of these things affect what's going on out there in the lake, in these big lakes. And it's important to keep in mind that these are complicated systems. And the way that we have to address this is we have to collect really enough data to start to understand these. So with that, I'll talk a little bit about our sampling program. So we do consistent sampling. It's ranged For the tightest range that we've done, it's daily for about 3 or 4 day periods. And the longest that we've done is about a 2-week interval where we're sampling every 2 weeks. What we do when we sample, we take an integrated photic zone water sample. So we integrate the entire photic zone of the water column. And then we take another water sample where we capture water from 1 meter from the bottom. In addition to that, annually, at the beginning of each season, we sample sediment with a ponar. We also filter water, and we collect those for DNA analysis, for both qPCR analysis and for metagenomic sequencing. We use a plankton net to do plankton counts and biomass, as has been described previously. We take field parameters every time we go out. We look at pH, dissolved oxygen, conductance, temperature, turbidity, dissolved CO2. We look at chlorophyll A and phycocyanin, and then we do second-disc transparencies. So no matter what, you always take your second-disc with you, no matter how much other stuff you're collecting. As far as our analytics, well, we do some pretty intense chemistry. We use the USGS National Water Quality Lab, and then also some boutique analysis that we like to call it boutique when it's somebody that does one analysis. Somebody does one analysis in our Wisconsin center, that's Pete Leneker. At the Water Quality Lab, we do full suites of dissolved and total nutrients. So we look at nitrogen, we look at nitrite, nitrate, ammonia, total nitrogen. We look at orthophosphate, and we look at total phosphorus. We also look at total metals and ion chemistry. Because we know that the blooms are affecting carbon dioxide in the water, that means that they're affecting the buffering capacity of the water. We know that, we see the pH changing, right? So that affects ion chemistry, and we think that there might be, there's some suggestions in the literature that some trace metal chemistry can actually play a role in how and when the organisms start to produce toxins. So we look at things like sulfate, chloride, alkalinity, iron, lithium, molybdenum, and zinc. All of those have been implicated as potentially being important for toxin production. In addition, we also look at dissolved organic carbon. So not unlike what Amber was just describing with flow cytometry, you can do the same thing on a bulk sample, and what that tells you about is you filter your sample, and then on that bulk sample, what you do is you look at it and you excite it with a UV light, and then it gives off some other UV light in response. And what that tells you about is the quality of the dissolved organic material that is present. The reason that's important is because as these blooms are growing and moving, we know that they're producing and exchanging molecules. And one of the things that we're interested in is understanding how the quality of organic carbon might be influencing either when they die or when they produce toxins. We do toxin chemistry. This is done mostly at our USGS Organic Geochemistry Research Lab, although we've done some work with the Michigan Department of Community Health as well. We typically use ELISA assays, which is the process that Amber just described. We look for microcystin LR equivalents. We do both totaled and filtered microcystin, and we do that pretty much on every sample. Now, at the beginning of this, back in 2014, we were really interested, we're going to find all these other toxins out there, and so we sampled for saxitoxin and antitoxin and cylindrospermopsin, and we never found any of them. We looked in all across all those sampling sites in the Great Lakes. The only place we found any other toxins was in Green Bay, and that actually wasn't USGS doing the analysis, that was the folks in Wisconsin that we were working with that found that. In addition, on some samples, we've done LCMS-MS. The reason this is important, you may or may not know this, the ELISA assays that we typically use are focused on this LR, but there are, I think, a growing list of 30 or more different congeners now of microcystin. Those congeners actually cause this test to respond differently. It's not a one-to-one response. So microcystin LR will give you a unit of 1. Other microcystin variants might, instead of giving you a unit of 1, give you a unit of 3, or a unit of 1/2, right? And because there's mixtures of these things in the blooms, we feel that it's important to get past just using the ELISAs and start to look at the individual congeners and to see how those come into play. To that end, Keith Lofton in our Kansas office is really interested in understanding the precursor molecules. Unlike a lot of proteins that bacteria make, these bacteria, these cyanobacteria, don't use this and package it in a ribosome. If you were here this morning, you might have heard about our DNA and our RNA. That's typically how the ribosome is how bacteria make proteins and fold proteins. Microsystems are assembled and folded extra-ribosomally, right? So they make these big molecules and then they fold outside of the ribosome, and that's kind of a unique process. So understanding these little precursor molecules might actually be important to understanding the system. Algal and related analysis. You've heard about a lot of this already, so I kind of dropped through this. We do microscopic cell counts with a contract lab. They do algal scans. They identify algae and cyanobacteria, and they enumerate cells per milliliter. The Great Lakes Science Center, where Marianne works, they do total biomass, ash-free dry mass, and chlorophyll analysis. We do have samples preserved to do phycocyanin extraction analysis, but we haven't found a very good method. So if anybody out there is doing phycocyanin analysis on extracted filters, I'd love to talk to you. And then microbiology, we're doing enumeration. For this we use quantitative polymerase chain reaction. I'm not going to get into the details of that, but suffice it to say that we can target individual pieces of DNA from particular organisms and then count those genes in the environment. We do that both for DNA, which is kind of an indication that it's there, and then for RNA, which is an indication that it's actively producing that gene in the environment. So we measure total cyanobacteria, total Anabaena, Planktothrix, total microcystis, and then we measure the toxin forms that produce microcystin of Anabaena, Planktothrix, and microcystis. And then we measure a gene for the production of saxitoxin and for anatoxin. As an example, this is just a gene diagram of the different complexes of the MCY gene. So, and then lastly, and maybe the most complicated thing to understand, I guess I left the best or the worst for last. So the Michigan Bacteriological Research Lab for the USGS, they characterize the bacterial community structure in sediment and in water. They basically, if you think about it this way, that I just described quantitative PCR as targeting an individual gene. What this sequencing process does is it basically targets every gene that's in the sample and sequences it all at once, and then you get back on the order of 3 million different sequences. You kind of have to sort through those with some advanced computing, but what that lets you know is it lets you start to understand the proportion of how things like Anabaena might relate to microcystis, might relate to Synechococcus, might relate to some of the other cyanos. And we're advancing that now to the point where we're including 18S into that, which will then tell us about how the things relate to green algae as well. All right, so we do all that data, right? And as you can imagine, we have this conversation a lot. We need more data. Yeah, we always need more data, right? We have that conversation a lot. But we're now to the point, like I said, where we're starting to analyze some of this. So I'll give you just a couple examples of the way that we're looking at this. One of our initial questions was whether the sediment might be seeding the blooms. So to orient you, Michigan, Lake Erie, Western Basin, Detroit River, there's that nice smile that Dr. Diz was pointing out. Maumee Bay, right there is the river raisin. That doesn't get talked about a lot, but it could also be an important source. So at the mouth of the Detroit River, what you'll see here is these are all collected on the same day. You see that microcystis is very low, Synechococcus is very relatively higher proportion, and then there's a bunch of total cyanobacteria that we're not even catching what they are. That has to do with the quality of the libraries we use. It turns out there are a lot of cyanobacteria out there that we don't have any way to compare to because they've never been sequenced before. So we have a lot of unknown cyanobacteria. Now compare that with Maumee, where you have microcystis in very high quantities in the sediment, right? So this is in the spring before there's any bloom activity. This is just a ponar grab of the sediment. And you see that the relative abundance of the cyanobacteria is greater. Compare that to the Toledo intake, which is right about there. And you see that it's different again. There's no Synechococcus here, but the abundance of cyanobacteria are very similar. And then you can see that there's diversity across the basin. So very complicated plot. This is my last one. So I'm right there. The thing I want to point out here, so these are dates, and those are the same three cyanobacterial species. So this is blue is microcystis, red is Synechococcus, which is a picoplankton, and then the green is total cyanobacteria. We have cyanobacteria qPCR data and microcystis qPCR data. So this is general cyanobacteria. That's microcystis specifically. And then we have the biovolume. So So one of the big differences that I want to point out, this is from W1 and W2, so these are on either side of the River Raisin. You'll notice that on let's see, where is it? It's the 721 sample to 8.4, we see this shift where Synechococcus drops off, right, and Microcystis shoots up. Here we see Microcystis come up, but Synechococcus doesn't go completely away. So even within this kind of closer spatial proximity, we're seeing differences in the Cyanobacteria community, and those tend to be related to things where we're seeing like large increases in biovolume. So what that's saying is that Microcystis is suddenly taking off. We're getting big increases in biovolume. And we have plots and plots of data with nutrients and carbon and all of those other things overlaid. Eventually, we're going to start to do some ordination analysis to really start to pick apart what we might be driving, 1, bloom formation, and 2, toxin formation. So here's some publications. James wins the publication thing. He's got 1 in press and 1 already out. The rest of us are still scrambling around in preparation. So I'd be happy to answer any questions.
About 7 minutes behind, so.
What does that mean?
Yeah, 7 minutes. Maybe one question while I'm getting the next
Anybody?
speaker's presentation.
Right. I'm happy. I can talk about this stuff all day. I love it.
You stumped them.
Yeah. All right. Well, I'll be around the rest of the day.
Thanks, Joe.